Good day, ma'am. Good day, classmates. I'm Karen Marie Galos, and the topic that I am going to discuss is about cell and cell division. Cell. What is cell? An animal's body is made up of millions of cells. Cells are the basic and generally the smallest part of the body that are capable of sustaining the process of life, metabolism, and reproduction. In biology, the smallest unit that can live on its own and that makes up all living organisms and the tissues of the body. An animal cell is the fundamental functional unit of life of animals. It is also the basic unit of reproduction. Animal cells were first observed in the 17th century when microscopy was invented. Robert Hooke, an English natural philosopher, was the first to describe microscopic pores, which he later called cells, albeit from the samples of a plant cork. Anton van Leeuwenhoek, a Dutch scientist, was also able to observe cells under the microscope, apart from the single-celled organisms such as prokaryotic cells and protozoa. He was the first to describe red blood cells and sperm cells of animals and humans. Parts of cell. Number 1, cell membrane. Holds and protects the contents of the cell. It serves as limiting membrane that separates the cell from the surrounding environment. It regulates the internal environment of the cells by regulating incoming and outgoing of materials. It transports substances in and out of the cells like nutrients, gases, hormones, and waste products. Number 2, cytoplasm. The space between the plasma membrane and the nucleus. It consists of semi-fluid ground substance. It also contains several kinds of structures called organelles. Number 3, nucleus. Control center usually located at the center of the cell. It is rounded, darkly stained structure separated from the cytoplasm by a double-walled nuclear envelope called nuclear membrane. Different kinds of organelles. Number 1, reticulum. Transport substances to all the different parts of the cell, a series of flattened interconnecting cavities and channels lined with membrane. There are 2 kinds of endoplasmic reticulum. Letter A, rough ER, with tiny particles of RNA attached to the membrane surface. Contains ribosomes rich in RNA that plays an important role in protein synthesis. B, smooth ER, tubular in appearance and does not contain ribosomes. Synthesize and transport lipids and steroids. Number 2, ribosomes. Small round structures which bind with mRNA and the site of protein synthesis. Number 3, Golgi apparatus, involved in transport of materials in and out of the cells. Number 4, mitochondria, sausage-shaped organelles, known as the powerhouse of the cell, which provides energy for all cell activities. Number 5, lysosomes, known as suicide bag because it contains hydrolytic enzymes needed in cell digestion. Number 6, peroxisome, similar to lysosomes in being membrane-bound sacs which contains enzymes responsible for production of hydrogen peroxide. Number 7, centrosome, spherical in shape containing 1 or 2 dark staining centrioles and plays an important role in mitosis. Number 8, vesicles, small cavities filled with either fluid or granular materials. Plays an important role in expelling excess water and some waste from the cell. Number 9, microfilaments, long, thin, tiny, cylindrical fibrils or tubules, predominantly of protein tubulin. Its function is form spindle fibers, skeletal elements of organelles like cilia, flagella, and components of centroids. Number 10, microtubules. Smaller than microfilaments and made up of protein molecules like actin. Its function is maintenance of cell shape and movement of inclusion and organelles within the cell. Parts of a cell. A cell is surrounded by a membrane which has receptors on the surface. A cell has several small structures inside it, including the nucleus, mitochondria, endoplasmic reticulum, and Golgi complex. These all have specific functions in the cell. Two types of cell. Number 1, somatic cell. Make up the bulk of the body and undergo mitosis. Number 2 is germinal and reproductive cells. The egg and sperm cells undergo mitosis, then meiosis. Cell division. Types of cell division. Amitosis, direct cell division. The cell simply constricts and finally divides into two daughter cells. Next is mitosis, incident cell division wherein the cell has to pass through several stages, namely prophase, metaphase, anaphase, and telophase, before finally dividing into two daughter cells. Stages of mitosis. Number 1, prophase. The nuclear membrane starts to break down. The nucleolus gradually disintegrates and disappears. The centrosomes or centrioles split and each centrioles repel each other, thus migrate towards the opposite poles, at the same time forming a centrodesmus between them from which the continuous spindle fibers originate. Some radiate outward, forming distinctive lines, the astral rays. Chromosome starts to condense, shorten, and thicken due to coiling of the major and minor coils, and thus become very distinct under the microscope near the end of prophase. The chromosomes go near the surface of the nuclear membrane. A sign for the final disintegration of the nuclear membrane. A sign for the final disintegration, numerous microtubules, the chromosomal spindle fibers originating from the centrioles, converge and connect to the kinetochore of the chromosomes. Number 2, prometaphase. The nuclear membrane and nucleolus are no longer visible. The centrioles have reached the opposite poles and the spindle fibers are completely formed. Chromosomes aggregate at the center of the cell in preparation for alignment. And number 3, metaphase. Chromosomes are aligned at the center of the equator of the cell. Number 4, anaphase. The centromere or kinetochore splits and active repulsion between sister centromeres occur. Thus, chromosomes are dragged and migrate towards the opposite poles. Microtubules, an interzonal or interchromosomal spindle fibers start to form and elongate between the migrating chromosomes. Mechanics responsible for the movement of chromosomes. The energy transmitted by the kinetochore of the chromosomes pushes the chromosomes towards the opposite poles. The elongation of the interzonal or interchromosomal fibers pushes the chromosomes towards the opposite poles. The shortening centromeres due to the removal of some monomers pull the chromosomes toward the opposite poles. Number 5, telophase. This is the reverse of prophase. Chromosomes have reached the opposite poles. The nuclear membrane is reoriented and reformed. The nucleolus and centrosomes are reformed or reappear. Chromosomes start to elongate, extend, and lengthen until the thread-like or filamentous appearance is assumed. Hence, the term chromosomal filament. A constriction of cleavage porous starts from the outer middle portion of the cells and through progressive constriction along a girdling cleavage furrow continues towards the center until it finally divides the cell into two daughter cells. Parts of nucleus. Number 1, nucleus membrane. Double-walled perforated outer wall membrane, the pores of which allow materials to pass either from the cytoplasm to the nucleus or vice versa, mainly made up of protein and fatty acids. Number 2, nucleoplasm or nuclear sap or karyolid. Gel-like nuclear sap in which are suspended the chromosomes and one or more nucleoli. Number 3, chromatin. Clumps of dense granular, thread-like network which are transformed into chromosomes during mitosis. Contains genes which carry genetic information necessary for replication and synthesis of protein. Composed of combination of protein, DNA, and some RNA. Number 4, nucleus. Dense spherical object which is largest in the interfaces. Consists of specialized areas of some chromosomes called nuclear organizer, ribosomal RNA and protein, and eats the probable precursors of ribosomal. Its function is construction of ribosomes, control protein synthesis, which ultimately leave and the nucleus and become organized in the ER. Functions of nucleus. Number 1, control center. Direct cell division since it contains all hereditary information in the forms of genes. Number 2, controls protein synthesis. Number 3, controls many metabolic activities of the cell. Note, those cells without a nucleus cannot undergo cell division, are not capable of protein synthesis, and are limited in their metabolic activities. And that ends my presentation. Thank you.